So before mitosis can occur in the cell, the cell has to replicate its DNA. There has to be a copy or two copies, identical copies of the DNA, one to go to one daughter cell and the other to go to the other daughter cell. So it makes a copy of itself during replication. So the DNA strand, if you look at right here, is two strands of nucleotides. So one's here and the other's here, and they're connected by their bases by hydrogen bonds. So we have to configure this DNA in a way so that it can be replicated. So we do this through enzymes. And there is an enzyme that unravels it, because it's normally in that helical sort of twisted together, right? So there's an enzyme that straightens it out, and then there's an enzyme, a special enzyme that takes the two strands apart or unzips it. Okay, this enzyme is called helicase, and helicase unzips the DNA, and it unzips it. It's already unzipped this side, it's unzipping it in this direction. So it's moving in that direction and unbreaking those hydrogen bonds and unzipping it. So once we have it's apart like that, we then have two different templates. There's a template here and a template here. So we're going to use these old strands to make the new strands to each one of those old strands is going to make a new strand, and that in itself, those two are going to combine to make the new DNA. So we're just going to match the nucleotides that are here. So if this is an A, then we have to bring in a T. If this is a C, we have to bring in a G. Okay. So there's two. Again, you see two different strands, two different templates. And if you remember before in the other video when we were talking about the structure of DNA, the two strands are not mirrors. The two strands run opposite one another. One of them runs 5' to 3' with its phosphate groups, and the other side runs 3' to 5' with their phosphate groups. So after the DNA strand is unzipped by the helicase, we have to have a starting point here. So we have another enzyme called primase. And what it does is it connects an RNA primer to the beginning. Okay, so this is the start of it, is that RNA primer. And then we have another enzyme called DNA polymerase. And it then starts to deposit the nucleotides here to make the strand. So it deposits the nucleotides, boom, 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 and we start making this strand. This strand on this side is running 3' to 5'. So it's basically following helicase. It's following helicase, and as a helicase is unzipping and opening up the DNA, it's adding nucleotides to it. So eventually it gets to the end and we'll have a whole new strand here. Okay, this strand, because it's just adding it and it's going to become in one shot the DNA strand, this is called the leading strand. Okay. On the other side, again, we have to start somewhere, and we start with an RNA primer. But because, again, this is going in the opposite direction, this is going 5' to 3', so we have to start inside the DNA, inside the strand. So we have our RNA primer, and we link it on to a nucleotide here. So here again is the RNA primer. This is the starting point. And then we get DNA polymerase, and we start adding nucleotides. So this is DNA polymerase. But you see, I'm going to run out of room here. I'm going to get to the end, and I had to start in this middle spot here. So as helicase is opening up the DNA molecule, then once this runs out here, then we have to start again with another RNA primer as it, like it say, opens up then to here. So now I'll start the RNA primer here. And then it runs back this way, adding nucleotides. And it does that over and over and over until we get to the end of the DNA. This thing here is called the lagging strand. And instead of making one big strand, it makes a bunch of little pieces of strand. And those pieces are actually called Okazaki fragments. And the Okazaki fragments are the pieces that get made, and the next piece will get made, and so on. And once that's done, then we have another enzyme called ligase that assembles the Okazaki fragments. There is another DNA polymerase called DNA polymerase II that replaces the RNA primers then with, so we have the RNA primer here, there's an RNA primer here. So we have another enzyme called DNA polymerase I. And that then switches out the RNA primer for a DNA nucleotide. So once that's done, then we get one strand of DNA here and another strand of DNA here. So this one was made complete. This DNA strand was assembled. So this type of replication is called semi-conservative. So it's semi-conservative replication. So that means that two templates create two separate strands. So each strand, each DNA strand, so we'll have use this color. So the DNA, there'll be one DNA strand here, and one new, and another DNA strand, an old and a new. So an old and a new and an old and a new. And these are exact identical copies. So that is replication.